Hello class, so today we will be going over arboreal mycorrhizal fungi benefiting mango trees in the field. Now to begin, arboreal mycorrhizal fungi are the type of mycorrhizal fungi that colonize the plant root cortex by actually penetrating the plant tissue and creating arbutules, which are arbor-like specimens of fungal growth. Now this differs from ectomycorrhizal fungi, which actually do not penetrate the plant's tissue. Arboreal mycorrhizal fungi, are the more common type of mycorrhizal fungi by far. Now to begin, we will go over our introduction. Now, mycorrhizal inoculations are found to be beneficial to fruit crops when applied in nursery in general because, as we know, they increase the range of the plant roots and allow them to take up micronutrients and water at a farther range that the roots can reach. And India is where this experiment was done, in southern India. The University of Bangalore is responsible for this one. They are the largest producer of mangoes, half of the world output, or actually 48% to be exact. And there are very few reports of fungal inoculation response in crops. So this field experiment is very beneficial and revolutionary, may I say. Now, early tests in nursery showed that improvement of pea uptake was noticed in pots that had fungal colonization, and I will go over why later. Now, to go along with this, there's an improvement in shoot pea content and plant growth in general, absorbed in these rootstock shoots, inoculated with some of the most common species here of Glomus massiae and Glomus fasciculatum. This is in pot culture, not in the soil. And so yeah, this study was taken up to evaluate the effect of inoculation of arboreal mycorrhizal fungi on establishment of rootstocks in the field and study effect of on plant growth and yield. So go to the abstract here. There was an examination of the rhizospheres of 3-year-old mango rootstocks for these arboreal mycorrhizal. AM is the for short abbreviation, just in case you hadn't noticed. There's the spore load and the root colonization at both 15 centimeter depths and 30 centimeter depths. The highest spore count was found at 15 centimeter depth. And if you go here, this just goes over the varieties that had the most, respectively. So the Totaputi being the highest. And so yeah, just the dominant genera of these AM was a Glomus for the most part, but also Acalospora. And yeah, Glomus fasciculatum and Glomus massiae are the most common. The most extensive colonization was found in the Totaputi rootstock, and these frequently occurring AM species were multiplied on finger millet, and these are fibrous roots of these monocot annuals, using soil right and plus with a 1:1 sand to soil mix. And so that's how they inoculated these fungi, so that they could place them into the medium and into the field later. And so all these mango rootstocks responded very positively to AM inoculation. It was noticed they had increased plant height, stem growth, and nutrient content, especially earlier with the pea content. Rootstocks with AM fungi also showed earlier shoot emergence. After 8 years, which is how long this experiment took place, the AM plants showed increased available P in soil, increased copper levels, increased zinc levels, more branches, as well as higher alkaline phosphatase activity. Now, alkaline phosphatase is actually given off by these fungi, and that breaks down complex molecules containing phosphorus into plant-available phosphorus in the soil. So that is very beneficial and very important for this experiment. Now, yeah, all these AM inoculum were easily multiplied on farm using the finger millet and applied annually. Now here we will go over methods and materials. So rhizosphere soil from 8 mango rootstocks collected at 15 and 30 centimeter depths is applied, as mentioned earlier, from 3-year-old plants. The spore count was determined per gram of dry soil, identified morphologically. Initial multiplication of finger millet was in paper cups with soil right, and then 12-inch pots with 1:1 sand soil mix. On-farm inoculation was conducted in a 4x4 meter field plot covered with polyethylene. Soil moisture was maintained at 50% field capacity. Now, the AM inoculum was applied at 5 centimeter depth for the finger millet. After 12 weeks, colonized roots were mixed with surrounding soil to produce bulk inoculum. Spores counted from pooled soil before field use. Polybags were filled with 400 grams of the 1:1 sand soil mixture and 100 grams of the inoculum of the fungi. Seedlings were planted, 10 replicates each for each cultivar, and we saw the cultivars were mentioned earlier, with Totaputi, Olord, Peach, those. And yeah, so the colonized plants, they're moved to sterilized pots with 250 grams of this inoculum. They were grown in glasshouse, which was kept between 14 and 32 Celsius, around 65% humidity, which is a high humidity, and this temperature translated into Fahrenheit for us is around 14. It would be around, let me think, maybe around 60 degrees, and 32 is around like 85 degrees. No, around 90 degrees. So pretty warm, relatively warm, good for mango growth. Growth parameters studied were the shoot and root length, dry weight, leaf phosphorus content, as well as zinc and copper content in the leaves. In the stems, I'm sorry. Field testing. The Totopodi seedlings were pre-colonized with this inoculum, as mentioned. They were planted with extra 250 grams inoculum and monitored for 8 years. Now after these 8 years, the dry weight, number of branches, and inflorescence time was recorded in comparison to the control group. Acid and alkaline phosphatase activity was measured with 410 nanometer absorbance, incubated in buffers with PNP substrate. Now next I will go over these interesting results. So the spore colonization was significantly greater at 15 centimeters, which shows that these arboreal mycorrhizal fungi prefer to inhabit the top layer of the soil, which makes perfect sense knowing our knowledge of plant roots and nutrients, which the O layer of the soil containing the most nutrients, and mycorrhizal fungi preferring to collect these nutrients from the plant, which prefers to collect these nutrients from the upper layer of the soil because that is where they concentrated. So this makes perfect sense. The highest colonization was in Totopodi and in the Veliculumban rootstocks. An area of 4 by 4 meters, they show that it can produce 5 tons of inoculum, which is very impressive and very, very promising for future crop growth. The inoculation significantly increased plant height, dry matter, nutrient content, and the root P levels. And this, as mentioned earlier, the fungi give off alkaline phosphatases, which increases plant available P content, which correlates with the higher root P content. Very beneficial for these plants, especially in tropical areas where phosphorus is normally the most deficient nutrient, in comparison to here, where it's arid and nitrogen is the most deficient. Now, overall, the yield significantly increased in the inoculated trees compared to the control, although I do not have specific data on the yield based on this research paper, other than it significantly increased compared to the control group, which is to be expected. And to add the graft shoots, these trees were grafted. The graft shoots were flushing 10 to 12 days earlier than the control groups, which is interesting, probably because of the increased P levels. The mycorrhizal plants with, this is a very interesting data here. The mycorrhizal plants with half P fertilization plus the arboreal mycorrhizal fungi performed the same as fully fertilized control trees with a fully fertilized phosphorus. So this just shows, well, you can eliminate a lot of rock phosphorus mining and a lot of expensive phosphorus fertilizer using these fungi. Very important. Now the mycorrhizal trees, as I mentioned earlier, had higher alkaline phosphatase activity, enhancing the soil P availability, as well as higher acid. Now, discussion-wise, these mango plants naturally host arboreal mycorrhizal fungi, as well as, in general, 80% of plant families worldwide do host arboreal mycorrhizal fungi, so this is to be expected. The problem comes when they are taken out of their zone where they have their native mycorrhizal fungi, and they must be inoculated, which was what happened here. Now, the inoculated Totopodi rootstocks led to faster graft establishment and enhanced growth, as we mentioned. And yeah, I mean, as we saw earlier, the different rootstocks do show different results in the AM colonization because different cultivars have different genotypes, slightly. The AM fungi improved phosphorus uptake, zinc uptake, copper uptake, phosphatase activity, enhancing P availability in the soil. We have done with that. Now, the mycorrhizal fungi possess high affinity for phosphate transporter genes as well. So overall, phosphorus is one of the nutrients that mycorrhizal fungi affect the most. And this is very important because phosphorus is a macronutrient in plants and is very commonly deficient, as I mentioned earlier. So this can be very beneficial in the tropical areas and really for any crop because 80% of plant families do have associations with mycorrhizal fungi. And really any plant can, because all they do is increase the range of the roots of the plant. They reach farther than the roots can get, and they have a symbiotic relationship where microbes of fungi, not being autotrophs, cannot produce their own sugars. So they get this from the plant, which can. And in turn, the plant gets nutrients, which it needs. So it's a perfect relationship. Now, conclusions. The mango rootstocks responded to AM inoculation in the nursery and the field. And this shows that you can use it in nursery as well as in the field, but in nursery. And it can produce in large quantities, as mentioned earlier, 5 tons per 4x4 meter area. And finger millet, which is a very easy, simple monocotyl to grow, to inoculate with. Yeah. Anyways, thank you for listening, and I hope this has given you some new information and considerations for the future.